So first of all, I would like to thank the organizers to invite me here to this excellent meeting. I have to admit I've never been in Hungary, but it will be certainly not for the last. And it was my pleasure to support Susanna yesterday with a thrilling driving to this very beautiful spot. As the title, it's a bit provocative, so I would like to share some of the current findings in our research group. But before I do so, I would like to express my special thanks to the what we call molecular pharmacology family. It's a growing family. We have several PhDs, as you can see. I'm one of the oldest one. I would like to highlight some of the research from current PhDs like Anna and Leshan, a former PhD who is now postdoc in Frankfurt, and a tenure tracker, Karim Rafi. He helped us a lot with virology and cryo-EM in the lab, and a dear colleague, Amalia Dolga. She's very specialized in neurodegeneration, so I can then sneaky peek also extend my knowledge here. And I would also like to say thanks to all the talented master and exchange students we frequently welcome in Groningen. So having said that, I would set the theme and would guide you first to one of the chronic diseases, Alzheimer's disease, which is characterized by progressive cell death. Alzheimer's disease, and these are here some numbers from America, is a major cause of dementia due to defects in the vascular vessels in the brain. The main problem in Alzheimer's is that we lose brain mass due to the occurrence of several cell death mechanisms. You see that here in this image that we have to deal with a loss in the hippocampal but also cortical area. Mechanisms which are certainly involved, but it's not limited to this, are effects on tau dysfunction, tau hyperphosphorylation, leading to a disruption in microtubules, and certainly we are fighting against plaques of the amyloid beta type, which are then leading to a dysfunction in neurons. When we focus on the risk factor for this disease, we certainly also should focus on pollutants, stressors from different sources out and indoor. And we focus our research on factors which are coming from outdoor environmental pollutants. Particularly, we focus on particulate matter coming in different sizes and thereby leading to the entrance in the body to different spots, either via introducing inhalation via the lung, but also other mechanisms are known. Particulate matter is a very aggressive part, also coming on with very aggressive compounds like nitrogen and carbon. And you see that it has been also shown here with labeled particles that in slices of the brain of postmortem patients, we really also can observe these pollutants in the brain. Now, particulate matter and the Alzheimer's disease, there is a clear link as in patients which are exposed to pollutants, there is a higher risk of cognitive impairment. And we also see in these patients typical changes like abnormalities in the brain structures and very typical Alzheimer's disease pathological hallmarks. We have a loss of brain mass, and the particles are either entering the brain via the olfactory bulb or via the blood-brain barrier, which is disrupted and dysfunctional in this patient. We get subsequently a change in protein stability, neuroinflammation, mitochondrial dysfunction, and oxidative stress. When we think about the mechanisms of degeneration or the loss in brain mass, one mechanism we are studying recently is ferroptosis. Ferroptosis is an iron-dependent cell death mechanism which is relying on the glutamate cysteine antiporter system, which is defining the glutathione content, so the red ox potential in cells. The GPX4 glutathione peroxidase 4 is central to that, leading to very aggressive polyunsaturated fatty acids, leading to reactive oxidation species and leading to holes in the membrane due to lipid peroxidation. We have shown in initial studies using an hippocampal cell line, HT22 cells, that an inducer or an inhibitor of the exchanger, the antiporter, that this is associated with cell death, shown here in this image of the cells, and that we could pharmacologically modulate this process by using an inhibitor, which you see here for the first time, CH3F4. It is an inhibitor of an exchange protein sensitive to cyclic AMP, EPAC proteins. I will talk a bit more about when I show you the data. For the studies in the upcoming period, we also used an inhibitor of GPX4 just to study the mechanism of lipid peroxidation and the underlying mechanism. We wanted to know whether pollutants, and we used NIST DEP particles. These are particular matter particles which are very well defined from a diesel engine, whether these particles induce per se cell death, and whether we are dealing here with ferroptotic cell death mechanisms. And of course, as we are molecular pharmacologists, we wanted to know the underlying mechanisms. Now, we defined with laser scanning, we defined the size of the particles. They are very small, so they should also enter the brain compartment. To our surprise, the cell viability, which was here measured with a simple colorimetric analysis, was not really impacted. Per se, the particles seemingly have no strong effect, so we searched further, and this was a work from Hong. And we observed that when we put a second hit, so not only stress the cells with the particles, but also inhibited GPX4, that you see here with the shrinking of the cells, that there's a profound cell death mechanism, which could be reversed by an inhibitor of ferroptosis ferrostatin 1. Now, to bring it to signaling, we first did LCMS analysis, and we analyzed the cyclic AMP content in the system. We observed fluctuations induced by the particles, but also RSS3, so this was a first hint to cyclic nucleotides or cyclic AMP, and we digged further. Here shown is that we analyzed the cell death mechanism by using propedium iodide. You have to believe me that the more purple we see, the more cell death. And you see again the second hit system that ISL3 and NISDEP induced a profound cell death mechanism, which was again sensitive to the inhibition of the EPAC1 protein CH3F4. Now, to guide you a bit through the mechanism, now I introduced already the ferroptosis signaling pathway driven by iron, centered to the GPX4, defining the redox potential in cells, proposed to lead to lipid peroxidation due to an increased reactive oxygen species production. On the other hand, we and others collected very good evidence that cyclic AMP modulation centered here with one of the proteins, EPAC, might be of particular interest. But please, I would like to guide the audience to the impact of mitochondria. Mitochondria are subcellular compartments which are a world on themselves. We have a cyclic AMP domain, we have calcium domains, and we and others were able to show that EPAC, particularly EPAC1, is highly expressed in mitochondria. So in the next part, we wanted to analyze a bit the interplay between cyclic AMP and calcium, as calcium overload also can lead to a death or unwell-being of the mitochondria, and we wanted to collect signs of lipid peroxidation. This study has been performed by Leshan, a current PhD student. She used fluor4AM, and again, I hope that you can agree that particularly when we hit the system with the two stressors, ISL3 and NISDEP, that we saw a profound increase in calcium, which was again sensitive to CS3F4. Here the quantification of the AM, the fluorescent intensity. And we also, as others had shown before, that calcium and cyclic AMP may be cooperatively regulated by a specific adenylyl cyclase, which is expressed in mitochondria. It's a soluble adenylyl cyclase. We also collected some pharmacological evidence that the soluble adenylyl cyclase is involved. Regarding the lipid peroxidation, what you can do, you can use fluorescent dyes like body pie. You have to believe me here again that the more purplish the color is, the more lipid peroxidation we have. Again, a process sensitive to CS3F4. As we do not want to rely our data only on fluorescent dyes, we also do biochemistry. So we analyzed the production of malondialdehyde, that is a reaction product of oxidized lipids, and you see here clearly that this is increased process sensitive to CS3F4. Now, to sum up this part, I could show you hopefully convincing data that particles are teaming up, can induce ferroptotic cell death, that this process involves some of the central players of cyclic AMP, EPAC, and that the calcium sensing seem to be also sensed by a specific adenylyl cyclase. Currently, we analyze to which degree the ER, mitochondrial communication, plays a role, as it is very important to avoid toxicity due to an overload in calcium, and there are some indications that particles specifically can regulate beta receptor function, so we are following up this on the molecular level. Now, Alzheimer, the disease, the symptoms are occurring, or the defects occurring long before a physician can diagnose a patient for Alzheimer. We see here the dysfunction in neuronal function and density, which is paralleled by an increased beta deposition. But here in green, we also see that immune cells of the brain, microglia, but also basic cells, astrocytes, seem to be activated. So we not only focus our studies on neurons, we also include microglia. Why we include microglia is shown here. Now, under the condition of a defective BBB, blood-brain barrier, which is a typical hallmark also of Alzheimer patient, there's an activation of astrocytes followed by an activation of microglia, which are in principle meant to clean up the brain, but on a certain moment in chronic disease, the microglia are saturated and are dysfunctional. This can lead to neuroinflammation and to the typical deep pathology. So what we did, we asked a very simple question: if particles are sensed by microglia, and if this can modulate their phenotypical features, as microglia can come in different phenotypes, they can act as anti-inflammatory microglia, but they can also produce a lot of very unhealthy components in the brain. So we are in the process still of the molecular mechanisms, but what we found is that exposure of the microglia, shown here from mice, that we see a morphological change in this microglia. This was accompanied by a typical activation sign of the microglia, which can be measured by for example, with IBA, which can be used as a sign of microglia activation. We not only analyzed the morphological change, we measured impaired phagocytosis activity using labeled thymosin. We have indication that there is a defect in lysosome function. We analyzed ROS production, particularly in the mitochondria, and we have evidence that there is an impaired AB degradation, which all explains that this is really teaming up together with the neurons to sum up to the typical AD pathology. A mouse is not a human, and at the end we want to treat human. So what we also do is we differentiate progenitor cells to microglia, and we were able to show that that under stress condition that we are dealing with the metabolic reprogramming phase, and we are now analyzing whether and to which extent cyclic AMP effectors like EPAC are involved. What we did is that we, by using this approach, that we could show that microglia derived from progenitor cells, human microglia, that they are indeed behave as our mouse microglia, as they show dysfunction, particularly in the lysosomal function, which is a sign of the fact that, the brain is less able to fight against aggregation of proteins. Now, I talked about the brain. I would now, in the last part of my presentation, I would now like to switch the gear, as we also do quite a lot of lung research. And I think everyone who's sitting here in the audience would expect that the huge majority of the particles are inhaled or entering the body via the lung. And this is indeed the case. The particles enter the bronchial part and the alveolar part of the lung, and they are expected to enter the systemic circulation due to the fact that the alveolar cells are dysfunctional and that the on top of the endothelial cell barrier is dysregulated, so the particles are really entering the systemic circulation. They induce systemic inflammation and can also induce, for example, dysfunctions in the heart. Again, mechanisms like protein instability, neuroinflammation, and mitochondrial dysfunction seem to be involved. Now, the lung, when it is damaged, and here an MRI image, and here some further illustration, a lung which is stressed either by pollutants or by smoke, you can easily imagine that in this quite sticky lung that we have problems with the airflow. We are fighting against inflammation, and particularly we have problems with the function also in the alveolar compartment. To zoom it in a bit more a bit more on the, not from the total lung, but on cell types. We do a lot of studies also in epithelial cells, bronchial epithelial cells, alveolar epithelial cells. Here you see that we, due to the collaboration, fruitful collaboration with the pathology in the hospital, we have access to primary epithelial cells. And we can analyze the effect of pollutants, and also as shown here for smoke. And we see that in healthy patients, so epithelial cells which you can cultivate from from non-smokers, that when we stain them, and you have to believe me that in green is one of our favorites, a protein which is keeping order in the cyclic AMP scenario, as we do not have only EPAC, we have pKa, we have ion channels, so someone has to safeguard the order. This is a protein we marked in green, and here in red, I assume that the audience is aware of the fact that epithelial cells express E-caterine. So caterines just to build up connections to their neighbors. And you see here beautifully in the primary cells that they have this cilia, so this is a happy cell. We also see that the proteins are co-localized. That was a reason of this study. But you also see easily, and even my mother, when I showed her this slide, believed me that smoke or stressors are not good for the cells, and that we have here a complete deorientation in this specific compartment. Here shown only a snapshot of what we also, for those which are interested, you can read the details in this article. This study has been performed by a double PhD degree or a double PhD student, Isabella Catani. She's now postdoc in America. She defended her thesis in Brazil and also in Groningen, and she was determined to analyze the effects of pollutants in lung cells. Here shown a set of data in which she could convincingly show in the lung cells that we are fighting with a mitochondrial dysfunction encompassed of a dysfunction in the organization of the mitochondria with a defect in cyclic AMP production, defects in alteration in the beta receptor functioning, potentially driven by a different subset of adenylyl cyclases. Also here we will analyze in particular the effect that particles have been shown to act as inverse, can act as agonistic allosteric modulators of GPCRs, so we are very fascinated by this aspect. But where I would like to bring you in the last few slides is to a model which has been used used in our group quite intensively. Here shown this work has been performed by Dan Lee, a PhD student. She defended her thesis beginning of this year. And what is shown, or what this illustration should give you a kind of a clue, that we are using also mini lungs. So we isolate epithelial cells, fibroblasts from mice and from human sources, and we cultivate them with a specific protocol, and then we analyze the function of 81, 82 cells and the effects of different stressors. The protocol we are using here shown for the usage when we isolate the cells, shown here for the simplified model of epithelial cells from the mice, and fibroblast cells we put together. We can analyze differentiation processes, proliferation processes in this very defined protocol, and we can analyze with, you see here this image, we have very small organoids and very large ones. The large ones are the alveolar type and the smaller ones are the airway type organoids. They have specific markers, ciliated cell marker on the airway type, and the surfactant protein C can be used as an alveolar type 2 marker. Now this study was ended also with a completely unexpected outcome, as we used LPS, what you see here, the major component of gram-negative bacteria. We aimed to use it as a stressor, but we ended up with a complete unexpected set of data I would like to share with you in the last few minutes I have. So we used this classical protocol, and we observed, and we didn't expect that, dose-dependently, that LPS did not ruin our organoids, but stimulated our organoids. As also Francesca said, you should be open also to unexpected results. Now we analyzed that so we analyzed the number, which was increased depending on the LPS concentration and also the size. We analyzed the type of organoids we have, and we found here, interestingly enough, that particular LPS had a profound effect on the alveolar type of organoids and in principle less on the airway type. Now, to bring it to the smart system is very nice, as we can also do the same for human. We can use human cells, and we here also obtained, at least for that the numbers of the human organoids were also increased. In the size, we didn't saw a very strong effect. We did RNA-seq analysis of the different cell types which are in these organoids, and several inflammatory markers and structural cells were changed But very profoundly, we saw an increase, a typical marker of the TNF-alpha receptor signaling pathway, and also factors like genes which are driven by NF-kappa B were profoundly changed. To sum that up, so we found in mouse and human lung organoids growth instead of what we expected, devastating or inhibiting effect. The effects of LPS were more on the epithelial cells in contrast to the fibroblasts. We saw an inflammatory gene expression and a tumor necrosis factor alpha signaling change. How could we, what are we working on? Now, TNF is a fascinating system. Here shown a system, now TNF1 and TNF2 receptor. We could think about the good and the bad guy. Normally TNF receptor 1 is envisioned as a bad guy, as survival signaling is normally inhibited. So at the moment, to be honest, we cannot explain the beneficial effect of TNF-alpha, but yeah, we still have to go, or I will still supervise several PhD students, so potentially we can unravel this miracle. And also in this model of the organoids, we are at the moment busy to study the role of air pollutants. Different viruses we also use as a stressor. And I'm happy to tell you more about the data. I thank you again, but particular thanks to all the students which are doing hard work and are quite an amazing, though sometimes also challenging team, and I'm open to any question.